Abstract.
Reactive oxygen species, WOS, generation is of intense interest because of its crucial role in many fields. Here we demonstrate that MOS-RQDs exhibit a promising capability for the generation of reactive oxygen species, which led to enhanced chemiluminescence. We discovered that the unique performance is due to hydroxyl radical activation increasing the active catalytic sites on molybdenum sulfide quantum dots, MOS-RQDs. The reactive oxygen species, such as hydroxyl radicals, UO, superoxide radicals, UOR,
and singlet oxygen 1O2 have been efficiently generated from H2O solution in alkaline conditions, in particular, the maximum
O yield was enhanced significantly, 9.18 times, compared to the Fe2, H2O2 Fenton system under neutral conditions. These findings not only enrich our understanding of the fascinating performance of MOS2QDs, but also provide a new pathway for ROS generation in all kinds of pH environment.
In this video, I'm going to talk about reactive oxygen species or ROS and how ROS is generated and what is the effect of ROS in our body, especially the harmful effect of ROS. So what are reactive oxygen species? So as we know, for our life, oxygen is really important. But this oxygen and some species of this oxygen could be very harmful. One of such species of oxygen is reactive oxygen species, or called ROS. And ROS are actually chemically reactive molecules which have oxygen. So these are chemically reactive molecules containing oxygen. Such ROS are peroxide, superoxide, and hydroxyl radical. So we would discuss them step by step. So here is oxygen, and this oxygen, once reduced by gaining one electron, it would form a superoxide radical, and superoxide radical is super duper dangerous. So this is the basic structure of oxygen molecule, and after reduction it would gain one electron and it would form a reactive oxygen species. As you can see, this is the stable structure of the oxygen molecule, but when it gains an extra electron, it is unstable. So in order to gain stability, it would try to donate the electron to others and make other molecule reactive. So in this respect, this oxygen plus one electron, which is actually superoxide radical, is very dangerous. Another super duper dangerous reactive oxygen species is hydroxyl radical, and hydroxyl radical have plenty of harmful effects on our body, and the enzymatic defense system which actually combat ROS cannot combat this hydrogen peroxide, this hydroxyl radical. So basically we need antioxidant to combat this hydroxyl radical.
Reactive oxygen species, WOS, is a collective term for oxygen-free radicals and molecules including superoxide radicals, UOR,
Hydroxyl radicals,
O, and singlet oxygen, 1O2, which possess high reactivity compared to molecular O2. Generation of abundant ROS is of immense interest in environmental and biological sciences. To date, the ROS are usually generated using a photocatalytic process via light-activated photosensitizers or a chemical process via ion-mediated Fenton reaction. However, the photocatalytic process suffers from undesirable biodamage by ultraviolet, UV, radiation and low reactive oxygen species production. This has restricted the role of photocatalysis for biomedical applications. The Fenton reaction can generate adequate ROS. However, this requires acidic conditions, pH equals 3-4, therefore, alternative reactions and conditions for ROS generation need to be pursued. Nanoscale molybdenum sulfides, MOS2, have gained widespread application in photoresponsive, energy storage and biosensor devices. Besides, MOS2 has been used as an excellent electrocatalyst for the hydrogen evolution reaction. However, the performance of molybdenum sulfide quantum dots, MOS2-QDs, toward chemiluminescence, CL, based on ROS generation has not been explored. Here, we demonstrate the excellent capability of MOS2-QDs to generate ROS from hydrogen peroxide in alkaline solution, which gives rise to Cl emission. Figure 1a, the hydroxyl radicals, O, from intrinsic reactions such as H2O2 in alkaline medium and the pt2, H2O2 Fenton system activate MOS2-QDs and generate active catalytic sites on their surface. These active sites then facilitate the conversion of H2O2 to generate a sufficient amount of ROS, which leads to a strong Cl emission.
So ROS is generated normally in our body. During oxidative phosphorylation, the complex 4, the terminal electron acceptor is oxygen. So oxygen accepts 4 electrons and form 2 molecules of water. Now, instead of accepting 4 electrons, if oxygen molecule accepts 1 electron, then it would convert it into a superoxide radical. So during a normal process in our body, some amount of ROS could be generated. Other steps like during respiratory burst by macrophages, ROS is also generated. So let's say our body is invaded by a pathogen, these red colored bacteria. So our macrophage and dendritic cell would take up those bacteria. So inside the macrophage, these bacteria have to be killed, and these macrophages kill these bacteria by means of ROS. Now during inflammation, ROS is also generated. So basically we have seen there are mainly 3 types of ways how endogenous ROS could be generated. So in exogenous pathway, by ionizing radiation such as UV radiation can damage our body and can produce a lot of ROS. Apart from UV radiation, tobacco smoke, like drugs and xenobiotics, and due to pollution, ROS could be also generated. By application of pesticides and consuming food and vegetables affected by pesticides, ROS could be generated in our body. And first most harmful effect of ROS is lipid peroxidation. So here you can see a plasma membrane, a plasma membrane bilayer, a lipid bilayer, and here we have drawn a hydroxyl radical, and hydroxyl radical has 1 electron extra. So it want to gain stability, and it can donate its electron to a nearby lipid molecule of the lipid bilayer, And then what happens, this particular lipid molecule get reactive, then it transfers its electron to the nearby, and then a chain reaction kind of thing takes place. And as a result, there is a potential damage in the cell membrane. And cell membrane is very important for functionality of the cell. Any damage in the cell membrane could cause osmolarity disbalance. When there is a osmolarity disbalance, there is a high chance that cell could be lysed. ROS can have potentially damaging effect on DNA. Like this hydroxyl radical, it could affect the DNA and it could cause a mutation. And if this mutation is not repaired, or this mutation is not repaired, there is a chance that it could be, and if this mutation is in the germ cell, then it could be segregated to next and next generation. And which could be potentially harmful, and it could be damaging. And apart from that, this DNA damage could lead to apoptosis also.
Conclusions. For the first time, we have demonstrated the excellent capability of MOS2-Gude or ROS generation. We explored that the synergistic effect of enhanced ROS production and ROS depletion were the main factors leading to the Cl emission of MOS2-Gude with H2O to an alkaline medium, and with the Fenton reagent under neutral and acidic conditions. These findings present a new pathway for ROS generation over the whole pH range. Thus, MOS2-Gudes have potential applications for degradation of organic pollutants and chemodynamic therapy. Conflicts of interest. There are no conflicts to declare.
Let's come to neurons. ROS could damage the myelin sheath of the neurons. As you can see in this neuron, the myelin sheath of the neuron is potentially damaged by the ROS. Thus, ROS can reduce the velocity of action potential transmission, and thereby it could reduce and it could decrease the neuronal functionality. So when there is a increase of ROS, risk of neurodegeneration also increases. And it is a highly classified research topic in these days. Apart from neuron, ROS can also have damaging effect on mitochondria. And once cytochrome C is leaked into the cytoplasm, cytochrome C could activate APOF1 and this APOF1 cytochrome C complex can actually activate other caspases, and there is a caspase cascade would go on, and ultimate fate would be the cell death, programmed cell death by apoptosis.